Thanks everyone for coming this afternoon to our distinguished lecture series. I'm standing in for Jeff, and as we all know, Jeff starts off by thanking me, so I'll thank myself. Thank you, Jay. You're welcome. And also thanking Global Water Futures for supporting the lecture series. It's a pleasure today to have Alex Konings come and give us a talk. Some of you in the Water Security Program got to speak with her a little bit earlier, and she was over in 121 for a good part of the morning. Alex has a great background with a bachelor's degree and a PhD from MIT, and a little break to move down to North Carolina in between, where she got her master's degree. And she's worked for some people that have been very influential in the field. Gabby Catul at Duke, Dara Entakabi at MIT, and then great postdoctoral experience as well at Columbia and JPL, where we overlapped but never really bumped into each other, even though we're in sort of overlapping circles. So Alex has done a tremendous amount of work over the past few years on, I guess we call it remote sensing of eco-hydrology. And today she's going to talk about remote sensing of plant water storage. And so it turns out to be a really important variable. We were talking yesterday, I guess I can't remember, it all blurs together. But I was involved in my early years before Grace took over my life, was doing a lot with soil moisture remote sensing. And we'd go out in the field and we would, that's my seat, but no, I'll sit over there. I'll sit next to John, sorry. Just save that one for me, okay, thank you. And that's my wife, by the way, okay, so careful. So where was I? Oh, yeah. Canopy water storage. So we're talking about the water in the plants. And it turns out to be a really important variable in going from the brightness temperature that we can measure, the microwave brightness temperature that we can measure from satellites, to actually inverting that into a soil moisture estimate. And the field experiment, so I was involved in the field experiment stage back before we had the soil moisture satellite, and we were testing out prototype instruments on planes, and we completely blew off the vegetation water content. We just did. It was really hard to measure. The NASA folks who were in charge of the experiments didn't want to deal with it, and we all knew that we were really injecting significant uncertainty into our estimates. So fast forward about 25 years, and here's Alex with some work on this that's, I think, ultimately going to be really, really important. So thanks very much, Alex, and thanks so much for making the trip out here. We know it's an ordeal.
Great. Well, I got here eventually, so it was fine. And thanks a lot for inviting me, Jay. I'm really excited to be at a place with such a strong reputation in hydrology, to be really one of the most sort of important places in both the history and it sounds like probably the future of the field. So Jay gave me a pretty nice introduction there. So I'll just briefly focus on my title slide here on giving due credit to all of the great collaborators I've been able to work with over the last couple of years. So Bill Anderegg at the University of Utah, Pierre Gentine at Columbia, and Park Williams right nearby at the Lamont Doherty Earth Observatory. And then especially to my group, so only a small fraction represented here, but Mustafa Momin, my former postdoc, did part of the work, as well as Krishna Rao, who's a current PhD student of mine. So they'll come up throughout the talk. So, we're talking here about remote sensing of plant water stress response through, as Jay said, vegetation water. And that might sound a little bit funky at first, because sort of the basic idea of plant responses to water stress in hydrology has been constant for several decades now, right? So the idea is plants have stomata, and those stomata allow the sort of uptake of CO2 that is necessary for photosynthesis. They also allow the diffusion of water outside, and they can open and close depending on conditions. And so you sort of, as a plant wanting to conserve water but also wanting to grow, are faced with basically a trade-off in how much you close or keep your stomata open under different conditions. And so there's kind of a trade-off there. We can parameterize that different ways. But for the longest time in hydrology, the representation of all of this has been, we say it's a function of soil moisture in some very semi-empirical way. If you actually look at what's happening with the plant physiology, the story's a little bit more complicated than that, right? Because the stomata are generally in the leaves, and there's a whole plant in between there and the soil. So water takes a finite amount of time to move from the soil through the plant canopy, and as it does so, it's mostly flowing through the xylem. The conductance of that xylem varies depending on basically how much water potential is in there. And so you actually have a whole continuum of water movement in the plant, all resulting in variations in basically the amount of leaf water potential in the leaves. And because of that continuum, because like I said, the xylem movement changes with time, and the balance of refilling and the amount of water you lose changes with time, you get a leaf water potential temporal variation that the plant is responding to that is very different from soil moisture. And you see that sort of in this figure in the bottom right here. So the top of that line here, for those of you looking on the right screen, is the evolution of soil water potential over the course of a couple of days. And then in the bottom here is the leaf water potential over the same number of days. And you see immediately that you have this really strong diurnal cycle that's just coming out from the diurnal cycle of evapotranspiration and the fact that it takes a while to refill that. But when you look a little bit more closely, you also see that at any given time of day, you actually see a slight decorrelation between the degree to which the leaf water potential is dropping and the degree to which the soil water potential is dropping. And so because the plant is not only doing things through stomata, but also has different xylem strategies, that decorrelation changes from plant to plant. And it's important because the plant is not actually responding to the soil water potential alone, it's also responding to the leaf water potential. Great, so we got this thing that's also been known for a while, but that hydrologists have ignored for a while. And I'd like to argue here in part today that it is important. And one person who's shown that before me is Gil Borer's group at Ohio State, as you can see in this slide. So I apologize, there's a lot of lines here, but I just want you to focus for a minute on the black line, which is the observed transpiration at a forest stand in northern New Jersey. This is over the course of about 3 months. At the same time, then, the different colored lines are different types of models for this transpiration. The red is sort of a classical Pem and Monteith approach that you might find in most hydrologic models. The blue we can ignore for now for simplicity, and then the pink is what happens when you actually account for all of these plant hydraulic processes that I was just talking about in the last slide. So, as you can see, the pink is much, much closer than the red. Accounting for that plant hydraulics is really useful, and we have work in my group in progress right now doing these kinds of tests at multiple different sites, and this is something that you really do see over and over again, especially during particularly dry conditions. And just for those of you who are not surface hydrologists, right, I'll just like to say and reiterate the idea that evapotranspiration is important to other parts of the hydrologic cycle as well, right? It will change the energy partitioning at the surface and possibly affect basically future climate through land-atmosphere interactions, as you sort of see in this schematic on the top here. And then it also, obviously propagates through to runoff by changing, how much water is, or how the water is partitioned. So to show kind of the magnitude of that intuitively, I've shown here a figure from a recent paper that's not quite about plant hydraulics, but hopefully you can bear with me in the analogy. So what's being shown in this figure is the results of several simulations that combine a Earth system model with a flood routing model propagating through the output of that sort of coupled land surface climate system that includes all of those interactions I just talked about. And you can just focus here on basically the blue line and the green line. And what the green line does is in that particular simulation, plants are not responding to changes of CO2 in the atmosphere. So it's a little bit different than plant hydraulics, but you could similarly say, hey, look, maybe we're getting the ET wrong in these models, so the plants there also would be transpiring incorrectly. And in this particular case, you're actually changing the high percentiles of the discharge by as much as a factor of 2. So who knows how similar that is for evapotranspiration? Or for plant hydraulics. I want to be clear here. But hopefully I convinced you in the last slide that plant hydraulics does seem to be important to getting the modeling right. And again, that's something that propagates even if you're not used to caring about ET. So hopefully that convinced you that hydraulics is important, and you could ask the question of, okay, well, how are we generally doing with hydraulics in our modeling? And the answer is not so well, right? I mentioned that most models don't even represent these processes. And insofar as they indirectly represent plant drought response through that stomatal closure semi-empirical function, the parameterization of that at large scales usually looks like something like this, right? So the idea is we say that vegetation in a given place is part of some sort of plant functional type. Most of you have probably seen this. And then as soon as you have a different plant functional type, that influences the quantitative parameters you use to capture the drought response. The problem is that that's a particularly bad assumption to make for plant hydraulics, as you see here. One sort of semi-intuitive reason for that would be just that even within a given grassland or a given type of forest, a given PFT, you often have multiple species growing side by side with totally different drought responses. And so you see that on the left here. This is one site in South Africa with, the exact metric doesn't matter, but basically the full range of different drought responses growing all at one tiny site. And then quantitatively, you also see that in the figure on the right here. So what's being shown here is the coefficient of variation of P50, which is a measure of that xylem conductance loss, and how that variation varies across different sort of groupings. And so if you just focus on the third and the fourth column here, we see in the third column the amount of variability on average within each plant functional type. And then in the last column, the amount of variability across the different plant functional type. And you see that the variability within plant functional types is actually higher than the amount of variability across plant functional types. So these basically are not providing you any information whatsoever about how that drought response is actually varying. So what do we need to do better in our hydrologic model and our hydrologic understanding altogether? Well, if we wanted to go out and do sort of the classical thing, we would go out and make a massive number of measurements of these same sorts of traits I've been talking about related to the xylem and the stomata. The problem with that is that there's a lot of species in the world, and it's very intensive to measure this. As you see here, right, this is the TRI database of plant functional traits in general. As massive amounts of efforts have gone into collecting all the data, or large fractions of the data that have been collected over time. These points are really large relative to the actual point scale that each of these measurements represents, right? And still, much of this map is gray. So not only is it basically impossible to get the right number of data from the ground up, to make matters worse, when we extrapolate from current on-the-ground measurements, we often do so in a biased way. And you can see that in this bottom figure here. So what this is actually showing here in the black is sort of an estimate of species richness across the world, across different latitudes on the x-axis. Not surprisingly, that richness peaks in the tropics. And then on top of that is the distribution of measurements from this map here at those same different latitudes. And you see that red distribution here peaks in the mid-latitudes, in places like North America and Europe that are close to the places where there's relatively more funding for science and where people can go easily take measurements. So that's one problem. But even if we had all of the in situ data in the world, if we wanted to put drought response in regional or large-scale models in a better way, we'd also have this problem of figuring out how to deal with all of those different species living alongside each other, right? What is the actual effective drought response of a given ecosystem at the types of scales that hydrologic models typically operate at? I personally like to posit that remote sensing provides the answer to both of these major challenges, right? It's inherently, at least if you use satellite data, inherently a global data set. And because it's not perfect resolution, it naturally averages over things. So this is just a figure of SMAP satellite, which I worked on a lot during my PhD, but pick your favorite NASA or remote sensing related image here. So what I'll do in the rest of this talk is I'll talk first about why I think remote sensing, not alone, but especially microwave remote sensing is useful for these kinds of problems. And then I'll sort of give a quick overview of 3 different studies that we've done over the last couple of years that show sort of the potential of these microwave remote sensing products for the types of questions that I just talked about. So let's delve right in, and basically I'll give you the spoiler alert that basically we use microwave remote sensing because it gives you a direct measurement of the amount of water in the vegetation. Why is that? Well, part of it is just sort of the physics. At the microwave frequencies, you're directly sensitive to the water content of the natural surface that you're looking at. And a second major advantage to microwave remote sensing, other than that it tells you the water content in the vegetation, is that it can see through clouds. And that makes it different from optical and hyperspectral and many other types of remote sensing. And that's something that's particularly important to questions about water stress response, right? And so just to really give you an intuitive feeling for that, I've shown here a map of the cloud fraction in July 2013. So this was a time when there was a big heat wave going on in Europe. I believe there was a drought in much of the Midwest and North America, if I'm remembering correctly. So it was a time that was particularly important for understanding plant responses to water stress and how they fit into the hydrologic cycle. And if we use any other type of remote sensing, this fraction shown on this slide of the time, we wouldn't have had data because of clouds. So the fact that microwave data can see through clouds is really a superpower in the context of looking at these sorts of questions. So just to do a little bit of housekeeping on microwave remote sensing. So today I'll be mostly talking about radiometer measurements, right, at these microwave frequencies. So what a radiometer does, it just looks at the passive gray body emission from the Earth's surface. I just want to give a quick shout out here to anyone who's particularly interested. There's a whole analogy here with radar instruments where we actually, send out an active measurement and looks at what comes back. And that has advantages and disadvantages, but you might come across this same concepts for radar in the literature as well. Like I said, there's analogies, and so basically, depending on whether you use radiometers or radars, you get the same sort of basic physics that's way oversimplified in this figure here, right? But what these radiometers do is they look at a signal from the soil, which is why, as Jay was alluding to in his introduction, these estimates or these data are usually used to estimate soil moisture. But as they sort of pass between the soil and the actual satellite, they pass through the vegetation layer. And that vegetation also has water and therefore attenuates the amplitude of the electromagnetic wave by a certain amount. That attenuation can be quantified in a quantity known as the vegetation optical depth, or VOD, and you'll hear me refer to that for the rest of this talk. So, like I said, and like Jay said, people have been doing this for soil moisture for a long time. Usually what they would do is they would, maybe when they're doing the field campaigns that Jay mentioned, try to do this a little bit, maybe in their algorithms try to account for this, but then they would throw away, for the most part, the data to get at the soil moisture. And so, again, the rest of this talk, I'd like to say, hey, look, don't throw this away. Let's try and use this to understand something about plants. All right, so if you know a lot about VOD, just a quick shout out. We use AMSRE data in most of these studies. It's not so important to following what's going on. So because we have all of these field campaigns that people have done for soil moisture, maybe imperfectly, as Jay said, but many, many years in a row, one piece of information that we've gathered over the years is that VOD seems to be basically linearly related to the total amount of water in the plant, which is why it's useful for us. And there's theory that sort of supports that linearity as well. So just for completeness, I do want to point out that, especially in the last 5, 6, 7 years, people have been starting to use VOD more and more. But most of what people do is they say VOD is a measure of biomass. And that's not a crazy assumption for people to make, right? You say, okay, well, if I have a certain amount of water in my plant, if I have more of that plant, I have the potential to have more water. And so indeed, as you see, in the slide here, offers redacted for the sake of anonymity, people make that assumption and do science based on that assumption all the time. This sort of was eating away at me for a while because it completely ignores the role of variations in water stress on that vegetation water content that VOD is sensitive to. And you see that just very simply in this equation here, right? We can look at as the VWC, the mass of water, just as the product of how much water you have per mass of biomass times the mass of biomass, right? Where that first ratio is just the relative water content. When we look at the plant physiological literature, at the laboratory scale, people have actually shown that relative water content is sensitive to the same leaf water potential that governs the plant hydraulics. So it seems that VOD should also be sensitive and potentially has, no pun intended, the potential to allow you to look at leaf water potential. So we have all kinds of different pieces of circumstantial evidence for this. I'll just skip straight to the most direct one. So my former postdoc, Mustafa Momin, what he's done here is he actually just went out and collected as much as he could data that other scientists had used for plant hydraulic studies in the field, and then just tried to compare it to VOD. And you see that here. So this is one summer at a deciduous forest in Missouri. We have this for several years and for several other sites as well. And you see it over the course of the summer here. So the VOD, you see as things dry out, decreases right alongside with leaf water potential, even as the leaf area index, the actual change in the biomass over the course of a summer, is not decreasing at all. So there's a relationship here. It's imperfect, in part because we're comparing basically 6 trees to a 25 by 25 kilometer pixel scale. But hopefully this gives you an idea that this is not a crazy sort of assumption to make. So I promised three different studies. So let's just jump right in. So the first of these is our approach to actually create a map of ecosystem scale variations in plant drought response or water stress response. And we do that through this particular metric that plant physiologists commonly use, known as the isohydricity. So what's isohydricity, you might ask? Isohydricity is the slope of the relationship between leaf water potential and soil water potential. And so you see that sort of shown in these examples here. If we have on the right, for example, a relatively isohydric species, right, what it'll do is it'll close its stomata relatively easy. It'll lose conductance in the xylem relatively slowly. And so as soil water potential dries down, or actually dries down this way. By the way, never mind. As it dries down over time, got slightly messed up by the negatives there. That leaf water potential is able to stay relatively constant, right? When instead we look at the oak sitting in this particular study, literally right next to the maple and experiencing the same sort of hydrometeorological conditions, it has different stomatal, different xylem behavior, And you see that over the same time period, the leaf water potential just drops. And so it's known as relatively anisohydric, right? So this is something that integrates both the stomata and the xylem traits that govern the plant drought response. And so I'll get just a little bit technical in the next slides and then be less technical in the rest of the talk, but I just want to be clear about all the assumptions here and show how we can use VOD. We can use that concept and basically get at that with our remote sensing data. So I said, this isohydricity, we can define it as sigma is a slope. And so one way you can think of it is just a number basically between 0 to 1 or a little bit more than 1 that you get from looking at the slope of a scatter plot relating leaf and soil water potential. And so to use the VOD to do this, we made a couple key assumptions. One assumption that we made that plant physiologists often make is we said, okay, at night, there's no transpiration. There's a lot of refilling of water. So after a long enough nighttime period, there's so much refilling of water that the leaf and the root zone soil water potential are actually in equilibrium. And so if we look at night, pre-dawn in plant physiology speak, or in our case, we assume that that's sort of done by 1:30 a.m., because that's when we get a satellite overpass. We make that assumption, and now we can use the VOD not just to look at leaf water potential, but to actually say something about soil water potential in the root zone, which itself is pretty cool because those same soil moisture data that I was mentioning earlier only really give you the top couple centimeters. So we use that assumption. We assume that all of this biomass that was in that equation earlier basically averages out if we look at diurnal variations, right? Over the course of a day, the biomass barely changes. So if we look at data, basically in the afternoon versus at night, it shouldn't matter. And then we make the assumption that VWC and leaf water potential are linearly related. That is linearizing a highly nonlinear process, but we kind of tried to see how much error we could get if we did that, and it's basically only 5%. So it's not as crazy as a lot of plant physiologists think it is when they first hear about us making this assumption. So you do all of that. I wanted to be clear about putting up these assumptions, but when you do all of that, the math works out really elegantly. And we get to a point where the linear relationship, the slope between observations of VOD at 1:30 p.m. and observations from the same satellite at 1:30 a.m. Is going to give us that same isohydricity, but now at the ecosystem scale. So we still don't know the exact relationship between VOD and leaf water potential, but in this particular case, that actual drought response metric of isohydricity is something that we can measure. And just to show you that this works for one sample pixel, right, I've shown here a pixel in India. This is the course of 1 year. We have data over 8 years. But you see the difference in the time series between the midday and sort of the middle of the night VOD data. And when we plot them together, we indeed get a slope that's a little bit less than 1. So you make these same scatter plots for all kinds of different pixels. Generally, you find a linear relationship like you would expect. So what does that look like? We put that together, and we get here a global map of isohydricity, a global map of drought response. The problem is this is a 25-kilometer resolution, and like I said, you usually have all kinds of different species that behave differently growing next to each other. So explicitly validating this is super hard. But we can look at this and sort of see if we see patterns that make sense, right? So for example, pines are known to be really isohydric. So we look at places like North Carolina, where I was for a bit. We can look at the south of France, places with a lot of pines seem to have really low values of sigma and seem to be mostly isohydric, as we expect. Same thing is true in the tropics, right? The bit of literature that has done these measurements there seems to show that they're very isohydric, exactly as we get with our estimate. And then last but not least, you also see the Corn Belt in the US and agricultural areas throughout the rest of the world showing up really sort of aggressively as really anisohydric places. And that makes a lot of sense if you remember that anisohydric plants are those that keep their stomata relatively open so that they can grow really fast. And so indeed, crops on the whole are bred to be on the anisohydric side. So aside from all these hand-wavy arguments, right, we can also do a little bit more sort of detailed validation using totally independent estimates. So I said isohydricity is something that's hard to validate because usually there's a diversity of species, but that's not true in agricultural areas, right, where you get these sort of monocultures cropping up. So what we can do is we can take a look at the US Corn Belt, where you have mostly corn and soybean growing, and we can take advantage of the fact that soybeans are known to be considerably more anisohydric than most sort of corn cultivars. And so now if we look at yield data, which is what's shown in the top row here, it's just an estimate of basically how much either soy or corn should be contributing to the effective isohydricity that we estimate at a given pixel. We see here in the bottom that if we compare the isohydricity data, which we get only from VOD, nothing to do with agriculture or yield data, and we compare that to sort of the spatial patterns of different differences in soy versus corn yield in different places, we get a pretty similar pattern using completely independent data. So that's pretty encouraging. Anyway, I'll also just briefly come back here and say, hey, if we actually look at the variation across the PFTs, right, coming back to what the models do of this data, it shows us exactly what we expected, which is basically that you have more variation within each of these PFTs for most of the PFTs than between them. And then you can take these data and look at all kinds of studies, or all kinds of different regions rather, to look at how this isohydricity affects ecosystem function. So we've done that, as you can see in these titles, mostly for photosynthesis so far. We could easily do it explicitly with evapotranspiration as well if we had the data. Unfortunately, ET data usually is pretty model dependent, and so it'd be a little bit circular to do that study. But hopefully, we're working in my group. We have some stuff in the works to move towards that anyway. So moving on to just show the breadth of potential of these data sets. They're a completely different application of the vegetation water content data. We can use it to estimate the drought-driven tree mortality rates. And this is work that's really led by my student, Krishna Rao, so I wanted to put his mostly smiling face up here, just give him due credit, because he's really pushed this forward. So why care about drought-driven tree mortality as a hydrologist? Might not be something that you've thought about much, but it is something that's happening more and more across the world. Unfortunately, it's a little bit difficult to actually come up with robust maps of drought-driven tree mortality events, but nevertheless, hopefully this map gives you an idea of how severe the problem is. So what's being shown here is sort of all the times when scientists heard about somewhere nearby some sort of tree mortality event, right? Really a case like this, not just a couple trees dying here and there, but really massive mortality at the landscape scale due to drought. And then each time that happened and some study was written about a particular event, that's a dot on this map. That's not an exhaustive list of tree mortality events whatsoever. It just happens to be the one that scientists were able to study. But it gives you an idea that these, just in the last couple decades, basically, as reflected by this map, that these are happening all over the world. And indeed, it doesn't just seem like it because there's more and more papers being written about this. We actually have evidence that this problem is getting worse. And you can see that in part from this study by Park Williams. So what he did is he was looking at a region in New Mexico, where there's been a lot of these mortality events over the years. And he looked at tree rings and showed, it's not shown on the slide, but he showed separately that this index that he came up with, this drought stress index from tree rings, is linked to how much drought-driven tree mortality happens. And then he was able to actually reconstruct that, or a pretty close match, right, of that index, which is shown in the red here, using only a combination of climate data as shown in the black line. And the sort of big ingredient in that climate formula of his, which had a couple of different terms, was the atmospheric humidity, the vapor pressure deficit during certain months of the year. Problem with vapor pressure deficit is that with rising temperatures and with sort of the basically pseudo-exponential relationship between temperature and how much moisture the atmosphere can hold, vapor pressure deficit has been increasing and is expected to continue to increase pretty dramatically with climate change. So when you put that through Park's formula and you calculate how much drought-driven tree mortality you expect in this region in the future, it's pretty bleak looking in terms of the trend. So one particular set of tree mortality events that has made the news a lot in recent years, especially down in California, is what happened in the Sierra Nevada in California during the recent drought there. So this is data from the US Forest Service, right? You can see the statistic here on the bottom, 100 million dead trees over the course of this drought. If you're like me, that number sounds like a lot, but might not mean much to you. But we can look at it instead as, the actual fractional mortality rate, which really is up in 2016, by 2016, almost at 7%. So that's, 6-7%, that's 1 in 15 trees in just 1 year alone, and this is happening over a couple of years and expected to, get worse with future drought. So this is a significant change to the land cover, which brings us to why should I care as a hydrologist? Well, if I'm changing the land cover that dramatically, I'm going to be, again, changing the partitioning of my water. So in California, a statistic that people throw around a lot that I haven't actually found a good source for, but that's a whole other story, is that approximately a third of sort of the water resources that are used seem to come from the actual snow melt in the same Sierra Nevada in the spring. And so you can imagine if we're changing the land cover by as much as I was showing in the last slide, that's going to change all kinds of different things related to the processes that govern that snowmelt and whether or not that snowmelt actually makes it to stream flow or other things that we can use. Things like changes to albedo, things like changes to interception, stromal conductance, and so on and so forth. And so you see in this particular study that adds up to a real hydrologic effect. So these are two different subbasins in the Sierra Nevada. This was a study particularly done with tree mortality in mind. And it just looked at what happened when we change large fractions of the canopy from trees to shrubs with different levels of LAI. And so you can see when we actually look at the sort of mean daily stream flow in the spring especially, you're really changing it by as much as 50%. So it's a little bit further removed from hydrology, but hopefully you believe me that it's still important to keep it in mind if you want your hydrologic models to be any good. Problem with tree mortality is that it seems to be really, really difficult to actually model. The sort of state-of-the-art plant physiological models still don't do that well in this respect. And you can see that in this figure here, right? So these are basically all of the state-of-the-art models and their predictions for two different variables that are the ones that people tend to link to tree mortality. On the left is how much of your xylem conductance you've lost. And on the right is the relative amount of non-structural carbohydrates. So basically the amount of sort of sugars available to the plant. And so this is at, again, a site in New Mexico, this place that is the single most well-studied place on Earth for tree mortality. And even here, when we look at these plots, right, the lines mostly disagree. Insofar as we have observations of non-structural carbohydrates, very few of these models seem to do a good job modeling it. And so in that respect, it's perhaps not too much of a surprise that it's really hard for these models to then actually get the mortality rates correctly. So, there's a talk about VOD. That begs the question, can we do something with VOD for tree mortality? And the good news is there's some really exciting sort of early data from our collaborators that suggests that the answer might be yes. Plant physiologists are used to thinking about leaf water potential, but Anna Sala at the University of Montana, a collaborator of ours, has shown in greenhouses that it seems like the mortality probability under drought of saplings, at least, is actually related to exactly what we see with VOD. It's related to that sort of relative water content of the plant. So we wanted to see if this was also true at the landscape scale, because if this is true at the landscape scale, that could be really useful for people trying to sort of just estimate where mortality is happening in the regions where we don't have data, or a little further along, if we can do some more research on this, maybe even try and sort of come up with early warning signs. So to do that in this particular study, what Krishna did is he said, well, look, all of these models, as I showed in the last slide, are really complicated. I don't want to try and build a model like that uses relative water content, because most of those models seem to disagree, and if I get a certain result, I will never know whether that was because my model was good or no good, or because of something about the data. So he just used a machine learning approach known as random forest. Sorry about the pun. But he used a random forest model to basically have the AI sort of build the best possible model of tree mortality in California during this drought, based on sort of airborne radar data of the fractional mortality, airborne visual data of fractional mortality rates. So we have this sort of optimal model from the random forest method, and then the advantage of having quote unquote an optimal model is then we can go back and see, of all the different inputs that we gave to this model, climate, vegetation, topography, there's a list of like 28 different models, does VOD actually make a difference, right? And so when we do that, step one is to see, okay, well, is our model any good? Can we learn anything from the contributors to that model? And the answer seems to be, well, in the grand scheme of things of mortality models, yes, when you do this, and you give it good training data and you use machine learning, it seems to work pretty well. So the first order, you see the spatial patterns between the observed and the predicted model match pretty well. This is, again, just to be clear, the fractional area of mortality in a given 25 kilometer pixel, so how much of that pixel actually died in a given year. And when we put the statistics together, you get an R-squared of about 0.6. That may not sound that great to you, but in the context of mortality studies, it's actually sort of better than a lot of state-of-the-art approaches. So we seem to trust this model. It's not a very practical one to use for actual estimation in the future, because we gave it a lot of training data. But again, we can go back and see if VOD made a difference. And in fact, it did, right? So if we look at the relative importance of all the different inputs that we used into this model, relative water content derived from VOD is the most important one. And not only is it the most useful for this model, it's more than twice as useful as the second most important input, which actually turns out to be, of all things, the elevation. By contrast, in this literature, people often use cumulative water deficit, which is just sort of a measure based on actual and potential evapotranspiration. And when you do that, you're down here. So these data seem like they could potentially be really useful in the future. Okay, so last but not least, I wanted to talk a little bit about a recent study we've done that allows us not just to map the drought sensitivity across the world, but to actually understand, or at least try to understand what might control it. So, to put that in the terms of what I've been talking about in the rest of this presentation, we can say we know that perhaps these PFTs are no good. We have an isohydricity map of some measure of drought response across different ecosystems. Why do these two maps not look the same? What governs those actual spatial patterns, if not PFTs? And so this is work that was really led by Bill Anderegg at Utah. So he deserves most of the credit for this, but I worked pretty closely with him on it. And just because we're sort of been talking for a while here, I'm just going to give you the preview of the outcome of the study right away and say that basically we find that forests that are hydraulically diverse, so that have a bunch of different trees with different hydraulic strategies, seem to be sort of more buffered to droughts, right? And the way that I think is really helpful to think about that is to think of it like having a diversified portfolio in the stock market, right? So you have a bunch of stocks. At any given time, the stock market might be up or down. Overall conditions might be more or less favorable, but any one stock is going to be doing a lot better than others. So some of your stocks might be going up, some of your stocks might be going down, and overall the best strategy is to average over lots of stocks. And basically, it seems like the same thing is true for forests. So how can you quantify that? Well, there's a bunch of different metrics that we looked at in this study. One is just to look at the relationship between evapotranspiration and things related to drought. So not just soil moisture, metrics related to drought, not just soil moisture, no groundwater, sorry Jay, we didn't have the right resolution for that, but soil moisture and vapor pressure deficit as our sort of atmospheric humidity metric. And then what we did is we went to sort of flux towers across the world where we had good information about the vegetation growing there and the conditions there. And we said, okay, well, how much of the observed evapotranspiration from these flux towers is actually controlled by these different variables related to water, right? So if we can't really explain the observed ET variability with these sort of drought metrics, then probably means that you're not that sensitive to drought, and vice versa. So we just looked at a simple linear relationship that we did at each site, and then what's the sort of coefficient of variation of that relationship. And so you see that here, right? You get a negative response, but a really significant in the grand scheme of things correlation with how much quote-unquote variation in hydraulic traits you have. So for this particular plot on the left here, this is just a standard deviation of that same P50, that same xylem metric that I've mentioned a couple of times now. And you see there's sort of a negative relationship with how sensitive you are to the drought. It's a noisy relationship, right? But it's a statistically significant one. And we don't expect this to be a perfect relationship anyway. The idea that any one thing would be the absolute determinant of how sensitive an ecosystem is to drought, is pretty crazy given the sort of range of processes involved. But there's a significant relationship here, and when you actually go and compare it to sort of other possible explanatory variables that explain different aspects of the different ecosystems at these flux towers, you get the same pattern over and over again. So this is the amount of explained variance for a whole bunch of different possible properties of these different forests. I'm not going to go through them in detail, but I just want to point out basically the bottom four here. So this is, again, that P50 and the hydraulic safety margin. The exact definitions aren't important, but these are two different metrics of basically how quickly you lose conductance in the xylem with drought. And indeed, both of these two very much related metrics, right? These two have the most explained variance of our drought coupling. And in both cases, the standard deviation explains much more the variance in the drought coupling than the mean does of that property. Even though, usually when we put things in models, we look at the means of variables, not their standard deviations. So this is really, really convincing analysis, mostly led by Bill that I worked with him on. But we have this problem still of this is 40 flux towers that were from an exhaustive literature search trying to gather enough information about the nearby species. It's hopefully not too difficult to pick out in the back of the room there, but when you look at kind of these black dots where these flux towers are located, almost all of them are in Europe and in North America, right? You got one in Africa, a couple in Australia, but basically we only really can believe these results in the mid-latitudes. So that begs the question, is this actually something that's real across the world? Is this something that's only true in certain climates? Is this a quirk of these particular 40 towers? Can we really figure out if this is true with remote sensing across the world? So to do that, I just wanted to put up the slide again to show you this equation again. So what we did is we used the VOD to basically come up with an analog of this equation. So again, we took advantage of the fact that in the mornings, VOD is something that is related to the root zone soil water potential, because at that point, the plant leaf and the root zones are in equilibrium. And then we used the sort of early morning VOD as well as the afternoon VOD as a metric of how much water you've lost through evaporation. And we used reanalysis VPD data to come up with something that's on the left here is sort of analogous to that drought coupling metric. And then we wanted to compare it to hydraulic diversity across the world. Problem is nobody knows how hydraulic diversity varies across the world. But at least at our 40 flux towers, it seemed to correlate pretty well with just the sheer species richness, just the sheer number of species growing at a site. So we used a pretty commonly used estimate of species richness across the world. And when you look at sort of some of these things that are super uncertain, you see, especially in the US, for example, some of the same spatial patterns. So this is super noisy because it's so uncertain, and because, again, even at the flux tower scales, this was a noisy relationship. But it nevertheless gives us some confidence that we see the same sort of effect of hydraulic diversity, both at the global remote sensing scale and at the flux tower scale. And we did a whole bunch of different supplementary analyses I'm not going to go through here that basically seem to confirm that this is not just some trick of the eye. When we actually use that same random forest trick that we used for the mortality estimation to see what's the most important explanation of this drought coupling metric here, it really did seem to be species richness and not sort of other vegetation properties. So with that, I think I'm probably coming up towards the end of my hour-long window here. I'm just going to leave up these take-home messages. The basic message that I want to just give is that plant hydraulics is something that is important to hydrology. It's making a comeback after kind of disappearing from view after the 80s. And it's a really, I think, opportune time for it to be making a comeback, at least for me, because with microwave remote sensing, we can really get an estimate of what the plant is doing and link that to plant hydraulics. So I showed you a couple different ways of doing that. It's not a perfect estimate by any means, and you got to be a little bit clever, but I'm pretty excited about how much more we can do with these types of data sets in the future. So thanks a lot for listening.
Well, thanks very much, Alex. Hey, before I forget, we have cookies and coffee, right outside, and we had it before. We'll have it afterwards if we didn't eat it all. And when it's all gone, then we'll go over to Boffin's for some beer and more conversation. Questions? John?
Alex, thank you for the talk. Really enjoyed it.
Great.
Oh, wait, microphone coming.
Very much enjoyed your talk, and prompted lots of ideas and thinking. But one thing, going to your global maps, the boreal forest jumps out in terms of its lack of isohydricity. And
Yeah.
Why is that?
It's a good point. I should have perhaps thought to give that disclaimer, should have seen that coming here in Saskatoon. It's not clear why that is. My suspicion is actually that we were not aggressive enough in filtering this data. So you see here, I've masked out sort of deserts and places of really low vegetation because it's hard to get at the tiny amount of VOD in those data sets. I think in these boreal areas, sort of similar things are happening. So, but
They're
on the
almost
side of
deserts in some ways. Yeah.
yeah, so there's very little vegetation, and then on top of that, what I also glossed over is that this sort of soil moisture VOD algorithm, that I showed the basic picture of, that stuff is imperfect. That's what I spent my PhD on, and it also completely breaks down anytime you have frozen soil or snow on the ground. So we've tried to filter for that in these calculations, but the chances that we did so, we basically use a temperature filter, and that doesn't correlate. Snow data, we've tried to put in some snow filters. The global snow data seems to be not good enough to do that robustly. So yeah, I don't know is the shorter version, but I think it's just because something got screwed up in the remote sensing along the way. So maybe in the future what I should do is grade that out in the map.
It's tricky. There's that period in the early summer when the roots can be frozen or partly frozen, and yet it's warm out, and so you get bone ratios of 20 or 30 over this thing. It's pretty crazy.
That is
even when you can see standing water around it.
Right. So that's a crazy number. So it's like, okay, well, how does that even fit into this framework? And then on top of that, figuring out when those time periods are actually happening, and whether or not they should go into this measurement a priori, we've just had some trouble with.
Yeah. And just one other question. When you're looking at the variability, towards the end, the spatial variability, I wonder, would thermal infrared remote sensing help with that? Because that canopy temperature is such an indicator of transpiration rate.
Yeah, that's a good question. It could. I'd have, and I think, like you said, it would come through the canopy temperature. So instinctively, I often sort of ignore that because I'm a little bit personally wary of all of the sort of modeling that people do when they go from the temperature to transpiration. But you should be able to just use the raw transpiration data, or the raw, temperature data, and just look at the spatial patterns in there. So that's an interesting idea. You'd have to think a little bit about maybe doing some like prism style detrending to try and take out other sources of variability that have nothing with the forest as well as you can, but I'm sure there's tricks to be played there. So yeah,
could be. Yeah.
That's great.
No more questions. John, you're shut off.
I'm done.
Ugly.
Thank you for your talk. Really interesting, the use of hydrocidity. I'm just curious, how do you account for stomata plasticity over time on the response to drought?
Good question. The answer is we don't. So this is the average over 8 years of data where we just wanted to get as robust an estimate of that slope as possible. So we just used all of the data we had. If you go, it's not cited in this slide, but if you go to our paper on this 2017 Global Change Biology paper that kind of introduces this data set, you'll see that we actually have a couple figures where where we calculate this not at the 8-year scale, but at the seasonal scale. And we look specifically about how this isohydricity seems to change between wet and dry seasons. And it's really interesting because in most places, like you would expect, you get basically more conservative, more isohydric in the dry season. And then, some tiny minority of points, actually the opposite happens. So we haven't really had time to go back and look into this further. But yeah, you could look at plasticity. It's just a matter of figuring out when you're starting to dip into the noise and figuring that out a priori.
Okay.
Yes, excellent talk, and lots
Great.
of food
Really
for
like
thought
that.
there. Thanks. I really like using the diurnal amplitude of the signal. I mean, you resolve or avoid a lot of problems
Right.
in using that. I
Yeah,
think
that's our only hope for getting this to work.
I'm really glad to see that approach being used here. Now, as I remember how plants work, leaf water content doesn't actually change very much between the extreme water potentials that a living leaf will experience. But the stem water content can change a lot. So
Right.
is a lot of your day-night VOD signal in the stem, and then it's related more to the, so either through capacitance in the
Yeah.
stem or through cavitation.
Right.
And is that what is really controlling things here?
I think that's probably right, but it's sort of too early days to really be sure. So I'm hoping actually this summer to go out and actually do, instead of having to scrounge together some other person's data, to actually go and do some, sort of stem water potential dendrometer and leaf water potential measurements at the same time to try and get at exactly the question you're getting at. But yeah, I mean, when we look at VOD, you're really seeing not just the leaves, but also the branches and also the stem, and it's sort of the effective variation in the relative water content of both of those. So insofar as that varies more in the stem, it's mostly the stem that's driving the signal. I used leaf water potential often when I'm talking about this, just because there's so many complexities that fitting anything of meaning into an hour otherwise gets pretty difficult. But you're right that that's a big uncertainty still, and definitely a complicating factor if you really want to understand these data correctly.
Well, that's right. And then understanding cavitation is really
Right,
important.
right. Yeah, and that's why
where-
you saw
where in
the
these trees it's irreversible, so that there's a strong link to mortality.
right. I was just about to say, right. That's why it works so well for mortality. It's not because of the leaves. That's absolutely because of the stems.
I think we have time for one more question.
Thank you. Thank you for your talk. Well, I noticed you mentioned other remote sensing products at the beginning of the presentation, like MODES. I'm wondering, is it applicable to use different remote sensing products to validate each other? Like you also mentioned the data sparse for validating work and.
Yeah, I mean, it depends a lot on the context, right? One nice thing about approaches like that is that even if all these other remote sensing data sets also have their own sources of uncertainty, right? I tried to be clear about ours. I didn't even scratch the surface. At least they're different sources of error, right? So you can do a little bit of that, but the devil is in the details always. So it depends exactly which data set you use, exactly what you're validating it with. But yeah, there's definitely some potential there. We don't really get at this metric with anything else, except maybe through ET, but that's a really heavily model-dependent process. So we haven't done it yet, but I'm sure there's absolutely settings for what you're suggesting is the right way to go.
Thank you.
Okay, well, thanks everyone. Really appreciate your coming, and thanks, Alex, for a great presentation. So Alex told us last night, was it the first 12 years that of your life that you lived in Belgium?
That's right.
So I've noticed with your accent, it's almost, it sounds very Canadian at times. And so
I'll
I
take that as a
thought maybe we
compliment.
could end by dubbing you an honorary Canadian, and thank you so much
Great. Well, thanks a lot.
for coming out. Let's continue our discussions outside over coffee and cookies. Thanks very much.